Stimuli in the body can sometimes be hard to localize. So one way that we can think about stimulus localization is to do what's known as a two-point discrimination test. This test involves measuring the minimum distance that's required to feel two distinct touch stimuli. So in other words, if you're not able to localize a stimulus well, then you'll feel 2 points of contact as only a single point of contact. But if you are able to localize a stimulus well, then you'll feel 2 points of contact as 2 points of contact. Now, there are several factors that can explain our ability to localize a stimulus differently, and we can use tests like this to evaluate that. So the first thing that matters for localization is the receptor fields. Receptor fields are unevenly distributed throughout the body. So in other words, some receptor fields have a punctate distribution. In places like your hands and your face, you'll find a lot of sensor units. Whereas in other places like your back and in your forearm and in your leg, you'll find fewer sensor units. And this is the reason why people can often very precisely identify the location of face or limb pain, but have a much harder time pinpointing abdominal pain or back pain. There's fewer sensory units there. The size of the sensor unit also matters quite a bit. On your face and on your hands, you have really large sensory units. The neurons there have a lot of sensory receptors at high density. And that's why, for example, if you have a hair on your face, it can be so annoying. But hair touching your back doesn't really bother you so much. Now, this example on this slide is talking about the same concept within the visual system. The receptors for color vision are highly concentrated at only 1 point on the retina on the back of your eye. And that means that we really don't collect a lot of information about color when light hits anywhere outside of this region known as the fovea. It's also the reason why it's difficult to resolve color when we're in really low lighting conditions. The light is more diffusely distributed across the retina, so we're collecting less information from these color receptors. Now, the size of a receptor field is also a factor, and this is a little bit counterintuitive because larger receptor fields actually lead to worse localization. When you have a large receptor field, regardless of where the stimulus is within it, it still produces an action potential in the same neuron. So on the left-hand side of this slide, you can see there's a receptor field labeled A, and that allows me to localize a stimulus to a much smaller area of the skin than receptor field B, which is much larger. Similarly, having overlap of receptor fields is also helpful for localizing a stimulus. On the right-hand side here, if neuron A fires an action potential, I know the stimulus must be somewhere within its receptor field. And if neuron B fires an action potential, I know that the stimulus must be somewhere within its receptor field. But if both A and B are firing the action potentials, then the stimulus can be localized to the area of overlap between those two receptor fields. In places like your face, where you have a lot of receptor fields, there's also a lot of overlap. And this mechanism actually becomes really important for proper stimulus localization. The final mechanism that's important for localizing a stimulus is known as lateral inhibition. This is a process in which the most strongly stimulated neurons essentially shut down sensory transmission in more weakly stimulated neurons. So this creates a situation where the central nervous system will receive one really intense signal as opposed to multiple more diffuse, weaker signals. So in the center image here, we're seeing a touch stimulus get relayed through 3 layers of neurons, a first order, a second order, and a third order neuron. There's a stimulus that's applied just above the first order sensory neuron, labeled B. But that stimulus is strong enough to also activate neurons A and C. Now, neuron B is going to have the greatest receptor potential. It's going to have the highest action potential frequency, at least in comparison to neurons A and C. And so if you take a look at the next layer, at the second order neuron, you can see that the neuron in the middle makes synapses with the adjacent neurons. And through this process of lateral inhibition, that neuron activates the postsynaptic neuron below it, but prevents the two adjacent neurons from activating their postsynaptic neuron. This process is known as lateral inhibition, and lateral inhibition often works through presynaptic inhibition, like we just talked about. Now, up to this point, in other units, we've always talked about axodendritic synapses, where are synapses between axons and dendrites. This synapse between the second order neurons is an axo-axonic synapse. That's a synapse between an axon terminal of one neuron and the axon terminal of another neuron. The left-hand diagram kind of recreates this scenario with these two presynaptic neurons that are in green and yellow, and the postsynaptic neuron is labeled in purple. So in this example, the most strongly stimulated neuron is the one in green, whereas the neuron in yellow is only weakly stimulated. And you can see that the neuron in green is synapsing with the axon terminal of the neuron that's in yellow. So when the neuron in green is firing the action potential, it can release an inhibitory neurotransmitter, an inhibitory transmitter you might have heard of before called GABA, onto the axon terminal of the neuron in yellow. What GABA does is prevent the voltage-gated calcium channels that are in that yellow neuron's axon terminal from opening. So even though the action potential is propagating in that neuron, the voltage-gated calcium channel in the axon terminal can't open, and therefore calcium can't enter and the neurotransmitter can't get released from that yellow presynaptic neuron onto the purple postsynaptic neuron. So in this situation, the only neuron that will be able to stimulate the postsynaptic neuron is the green one.